In this video, we will be talking about cell lysis, which is the process of breaking cells apart. The main purpose of cell lysis is to extract intracellular components, including proteins, DNA, or other organelles. This is necessary because oftentimes products grown inside the cell need to be separated from the cell membrane. There are two main methods of breaking cells apart: mechanical and non-mechanical. Mechanical methods usually involve force and generally require a machine to physically break apart the cell. Non-mechanical methods generally involve chemicals and reagents to break apart the cell at the molecular level.
The first mechanical method we'll be discussing is arguably the most simple. A sample is placed into a mortar and ground down by a pestle until the sample is sufficiently separated. The mortar and pestle method is most often used on a very small scale where it is very fast and convenient. The downfall of using a mortar and pestle is that it often damages the intracellular organelles. This is only a problem if these are the desired products. Another mechanical method similar to the mortar and pestle is bead beading. Bead beading uses a rotary mill full of either glass, ceramic, or steel beads. Samples are placed into a mill where the beads break down the cell membrane as the mill spins. The sample is collected after the mill completes its cycle. Another mechanical method is sonication, which uses a machine that outputs very high-frequency sound waves. These sound waves work to break down cell components. In practice, a sample of cells, bacteria, spores, or tissue are often placed in an ice bath to prevent heating and subsequent denaturation of proteins. An example is seen at the bottom of the screen. Another very common method to perform cell lysis is homogenization. Cells are forced through a very small opening, which strips them of their cell wall and separates intracellular components. This image is a version of the French press, which showcases a solution inside that is running at the end of the apparatus as the plunger is forced down. The final common method of mechanical cell lysis is freeze-thaw. Cell walls are broken down by repeatedly freezing and thawing a sample. Ice crystals form within the cell and cause the cell to rupture. The downfall of this method is the large amount of time it takes to finally open up a cell.
There are three non-mechanical ways to break open a cell. The first is osmotic shock. This is where the cells are placed into a saturated salt solution which induces osmosis. The water then either rushes into the cell, causing it to swell and explode, or water rushes out of the cell, causing it to shrivel, which is shown in the diagram below. The second non-mechanical method is the use of detergents. Detergents are water-soluble molecules that bind to the hydrophobic tail of the lipid bilayer. The detergent then peels the membrane apart, leaving gaps for the intracellular components to leave the cell. The typical process for a detergent is represented in the diagram below. The third non-mechanical way to rupture cells is with enzymes. Each enzyme is specifically chosen to break down a certain cell membrane. For example, cellulase is used to break down cellulose, which is the membrane of plant cells. This allows the proteins and polysaccharides to be released from the cell. A picture of cellulase is shown below.
As with anything, there are pros and cons to each method. Mechanical methods can handle larger sample sizes and are significantly less expensive than non-mechanical methods. However, they take more time and are much harsher on the cells than non-mechanical methods. Non-mechanical methods can target specific cells and require less time and materials than mechanical methods, but are generally much more expensive. The best method of cell lysis depends on what the goal is. For example, if a company is trying to extract as much product as possible, as cheaply as possible, a mechanical method is best suited for them because they are generally able to handle larger sample sizes and cost less than non-mechanical methods. But if the desired product is difficult to extract, non-mechanical methods would be better because they can target specific cells and leave other organelles intact. That pretty much does it for cell lysis. Thank you for watching.